All right, thanks, Caroline. Welcome, everyone. So our project contains of two components. The first one is using genetic tools to estimate the impact of vector control on mosquito effective population size. And then more recently, we added a second component that is focused on characterizing metabolic resistance in Houston populations of Culex quinquefasciatus and Aedes albopictus. So in the first component of our project, we try to answer these questions. Specifically, what we're most interested in is what impact do sort of organized vector control efforts have on mosquito population size. So the goal here is to estimate or infer the demographic history of populations in Culex quinquefasciatus population in Houston and compare regions that have received extensive treatment versus regions that have not. This kind of analysis might also provide some insight in what kind of bottleneck these populations experience during the seasons when they're less abundant. And our study time frame also includes Hurricane Harvey, so we might also be able to infer something about how Hurricane Harvey impacted mosquito populations. And this work will be done using a method called approximate Bayesian computation. So we just want to explain briefly how that works. And essentially what the main, the principal idea behind this is that you get a population genetic data set from the population that you're interested in, and you need multiple time points across the time frame that you're interested in. And then you define various demographic scenarios that you think might explain your, might reflect the true history. So for example, one demographic scenario might be a constant population size over time. Another one might be a bottleneck or an expansion or fluctuating sizes. And for each of these demographic scenarios, you simulate millions of data sets. Okay? And these millions of data sets can then be compared to the actual data set. And that allows you to derive posterior probabilities for each scenario and hopefully infer what is the most likely actual scenario. Now, when you do these simulations, for each scenario, the parameter values or the priors that you put in for things like population size and the timing of events or timing of bottlenecks, for example, varies for each individual simulation. So once you have identified the most likely demographic scenario, you can look at the simulations of that particular scenario, take the top 1% of those that are most similar to your actual data set and look at the parameter values that were used to simulate those 1%. And that allows you to infer things like effective population size and timing of bottlenecks or expansions, for example. And so the inspiration for this work was really previous work that we've done in Equatorial Guinea. So I want to briefly go over that really quick so that you can get an idea of the sort of possibilities of these tools. So in Equatorial Guinea, we looked at 7 populations on both Bioko Island and the mainland, including several species of malaria mosquitoes that were controlled either through IRS or LLINs in really organized control campaigns. And we got time points from the start of the control to several time points afterwards. And really what we wanted to ask in this particular study was like, what impact did the start of organized vector control have on the effective population size or the genetic size of these mosquito populations? And I'm going to just show you one result as an example. So this is from a village in Yenge on the mainland Equatorial Guinea. And here we found that the bottleneck scenario in which there was a sharp reduction in population size at the highest probability with 98% posterior probability. So really quite high. And then we looked at the parameter values that were used to simulate the top 1% of these simulations. And that allowed us to look at the parameter values or the NE values that might have reflect the population before and after the intervention. So we see really quite nice peaks here. So the pre-intervention size, the genetic size was estimated at 13,200 and the post-intervention size was estimated at 1,900. So an 85% reduction in the population size. And then we looked at, okay, when did this population size reduction occur? And it occurred about 88 generations ago, which matches exactly the start of these vector control campaigns. And so with that, we were able to show that in 6 out of the 7 study populations, we saw a really sharp bottleneck, and that IRS reduced population size by about 80 to 85%, whereas the distribution of LLINs, long-lasting insecticidal nets, reduced the population size by about 55%. We also included control populations, one from Mali and one from Cameroon, so negative control populations in which there was no organized control effort. And there we did not see any evidence of a population bottleneck. And so that raised the next question. So what happens during individual spray rounds? And so we focused next on a single population from Punta Europa on the northwestern point of Bioko Island. We had 6 time points that were interspersed with 2 spray rounds, okay? And we did the same analysis, and we found that this particular scenario had a very high posterior probability, 97%, so really well supported. And then these are the parameter values of the population size during March and May, and we see that it's a sharp decline during May and July when there was a spray round. But then we see that within 4 to 5 months, the populations fully recover. It's only to be knocked down again by the next spray round. So this kind of showed that the insecticide did not have sufficient residual effect, or that in order to really knock these populations down on a sort of a consistent manner, we really need to do more spray rounds rather than every 6 to 12 months. So this work showed that we could get pretty good resolution of demographic history on even very short time frames. So this was really the inspiration for the work that we're doing in Harris County. So we're focusing on 2 areas, Area 415 and Area 802. Area 802 saw a lot of organized control by Harris County Public Health, whereas Area 415 did not, okay? And so we have 4 sampling points from 415. We have also 4 for 217, and that includes Hurricane Harvey. Okay. And so the goal here is really to sort of to estimate demographic scenarios, compare 415 to 802, and see if these extensive control efforts really affect the demographic history of these populations. So we're still working on this. So we have the data set, and we've started data analysis as well, which is a very lengthy process because there's a lot of determining the proper prior distribution that goes into doing these analysis. And every time you run an analysis, it takes many, many days for it to be completed. Our preliminary analysis so far of the 2016 data has not yet shown very good resolution to distinguish between the various demographic scenarios. So they already know that affected population sizes of these mosquitoes may be very, very large, and that could be influencing our ability to detect demographic scenarios. So things that we're exploring now is that time points may be too close together for good resolution based on the population size. So we're combining 2016 and 2017 data and reduce the number of time points. We're also exploring the option that we haven't run enough simulations yet, so we're increasing that. We're working on the various prior distributions and trying to get both the shape of the distribution correct, but also the range. And the other option might be that we haven't obtained enough population data. So for each of these time points, we analyzed 100 mosquitoes, I should say 96 mosquitoes for 15 microsatellite loci, which for population genetic data set is pretty good sample size, but for this kind of approach, it may not be sufficient yet. So, if nothing else works, we'll explore that possibility. Now, the second project that we're working on is something that we've added more recently. This is characterizing metabolic resistance in Houston populations of both Quincus fasciatus and Aedes albopictus. And so there's various insecticide resistance mechanisms, as I'm sure you're all aware. So metabolic resistance refers to essentially the upregulation of various genes that might break down or sequester insecticides. And so 4 gene families that are particularly implicated that are the cytochrome P450s, esterases, and then the GSTs. And so those would be things that we'll be focusing on specifically. And like our previous work, this is in close collaboration with Harris County Public Health. We couldn't do any of this work without them. And so we designed 2 experiments. In one experiment, we are collecting Culex quinquefasciatus from the field. We're exposing them to malathion bottle assays and then separate them in those that are resistant and susceptible. And these are stored for RNA-seq, essentially whole transcriptome sequencing. And we obtain 6 reps, and we have each rep consisting of 10 individuals that are pooled. And then we we subject these to sequencing and compare the expression of the detoxification enzyme. So the question here is that whether, susceptible mosquitoes have lower level of the expression of some of these genes than resistant mosquitoes, either through constitutive expression or because they're able to upregulate their expression more quickly in response to exposure. The second experiment is aimed at looking at seeing if mosquito population Houston have constitutively higher expression of detoxification enzyme. So what we're doing here is just taking Quincke fasciatus from field collected eggs, rearing them in the lab, and also have a susceptible Quincke fasciatus colony, and we take samples from those as well. Again, do whole transcriptome sequencing and compare the expression of these genes. And so the expectation is that this will provide a list of candidate genes that then can be compared to either what's known in the published literature, whether these genes have been specifically implicated in insecticide resistance already. Otherwise, we'll do a RNAi to validate the gene function by essentially knocking down the expression of these genes and see if that affects the susceptibility to resistance to insecticides. So where we are right now with these is that the QLEX Quincke fasciatus samples have, we've done Houston have completed the assays. They've provided us the samples. We've submitted them for sequencing after screening for, after testing for species diagnostics and all that. And I just checked with the sequencing facility. They've had them for a while, but they're actually going to put them on the machine tomorrow, and we should have that data at the early part of next week. In terms of Aedes albopictus, eggs have been collected during the summer of 2020, but then COVID-19 derailed everything, and so these still need to be tested for. We still need to, or Harris County still needs to do the bottle assays before they can send us these samples. But once we have them, we can process them fairly quickly and obtain the RNA-seq data in short order. So I don't have any specific results to share right now, but this is sort of the update on where we are right now. So this work is being done by a Texas A&M PhD student, Xingyu Wang. And of course, many thanks to collaborators as Harris County Public Health that are really important for this project. We couldn't do any of this without them. And thank you.
Thank you, Dr. Slotman. All right, just a reminder that we are holding questions till the end of the session for these speakers. And up next, we have Dr. Gabe Hamer, who's already shared his screen. Wonderful.
There we go. I'm unmuted. When I just shared, did you see the big one or the?
We saw a bigger version. I think if you click that button, that's fine. We can all see.
You see the full screen, right? Or
yes.
Okay.
Actually, we're seeing the presenter screen.
All right.
Oh, okay.
Let me just share a different one here.
Sorry.
There
we go.
Okay. All right, I'll advance. So yeah, I'm going to highlight a few, roughly three projects we've been involved with regard to vector control. One is the autocytogravid trap. So that would involve principally Willie Juarez, and a lot of others helped with that in South Texas. I'll talk about our partnership with Harris County and looking at their adulticide data to see how it impacted Culex abundance and control. So a lot of people to thank there. And then I'll wrap up with our human survey project, which involved Katie Dickinson and Natalie Benacos at Colorado School of Public Health, and they led that effort. All right, so just a quick reminder, when it comes to integrative pest management or mosquito management, obviously there's a lot of control tools to use, but the important thing is to really evaluate and judge whether or not those tools are working. And if they are, great. If not, obviously we need to reconsider. Also, as we all know, there's a lot of innovative and emerging techniques and tools and approaches to help control vector-borne diseases. So that's one of the goals of this project broadly, is to kind of help advance the evaluation and help scale up some of these options. So as a reminder, this is a slide that captures the different study designs that can be used to evaluate vector control tools. There are some study designs, especially, for example, operational control, where there's no control group. Those are not recommended. There are a number of observational or non-randomized control trials. These are often what are done during, operational vector control. So sometimes you can gather sufficient data to help evaluate vector control. And then, of course, what's considered the gold standard would be a randomized control trial. So I just wanted to highlight a few examples of these that our projects have been involved with. One is just our partnership with Dallas. So we did publish a perspective looking at the distribution of Culex mosquitoes infected with West Nile during that 2012 epidemic. So many of you remember the Dallas-Fort Worth Metroplex had a lot of West Nile in 2012. And so this only publishes the kind of a risk map of what the probability of infection looked like across that county in that year. When we started this partnership with Harris County, Dallas County, we were interested in seeing if the aerial adulticide campaign, which occurred that year, and it doesn't often occur up there, we wanted to see if that intervention had an impact on Culex abundance or infection. But it turns out there were very few, if any, mosquito traps in areas that did not receive the aerial adulticide campaign. So as a result, that would have been one of these categories down here where that would have been a scenario where we did not have a control group, and thus those are not recommended studies for evaluating vector control. We also have other examples of partnerships. So this is a nice one with the city of Brownsville. So in 2017, we all know Zika was still a major concern, and a lot of transmission of Zika virus was occurring in northern Mexico in 2017. And so given the concern of local transmission occurring in South Texas, the state helped produce some emergency funds, and then that allowed Clark Mosquito Control to come in and do a BTI-based larvicide campaign in the city of Brownsville. So a lot of people were involved with this. Whitney Qualls from the State Health Department, the city of Brownsville, and even John Paul Mutebi from the CDC has been working with Brownsville for a long time. And I think this is a great example of an operational control activity. It was done in an emergency situation, but thanks to our center existing, I think we help provide some of the motivation and then also the capacity, the personnel, the lab, and the people to help really evaluate, did this intervention work? And so we did publish that last year. So again, a perfect example of how this center existing helps us create these partnerships in real time and produce good evaluation pieces. And then I just want to then focus a little bit more on the Harris County effort. Again, another observational data set, a very large one, looking at their adulticide use and its impact on Culex abundance and infection. And then kind of the representative of this top of the triangle, which is a randomized control trial we did in South Texas using the autocytogravidova trap. So that's what this study is. A lot of you are familiar with the AGO or AGO, the autocytogravidova trap. So Roberto Barrera and his team in Puerto Rico helped develop this trap. The idea is that it's a 5-gallon bucket with water. Female mosquitoes enter trying to lay eggs. They're not able to access the water, and while in the container, they come into contact with the glue board, get stuck, and they die. So this is a nice surveillance tool. So it can be used to monitor female Aedes aegypti abundance. But then also, if you deploy enough of these, they can also be a control tool. So in South Texas, we worked in the communities, 8 communities that stretched from McAllen on over to La Feria. So in 2016 and the start of 2017, we started just baseline surveillance. And then in August, we deployed 425 of these agos in randomly identified 4 of those communities. And then we did it for a few months, and then we waited, we pulled them, and then deployed them again in the other communities in August of 2018. So quickly, just highlighting some of the results. So overall, we saw very little evidence of suppression in 2017. We did see evidence of suppression in 2018. So we think one thing that happened, and keep in mind, really the only form of vector control that was done in these communities was this AGO. So there wasn't source reduction, no adulticides, et cetera. But in 2017, our community coverage, so the number of AGOs deployed per home was not that high. So we were only at about 51 or 57% of the homes had 3 AGOs. We were hoping to get to 80%. So then after additional recruitment and engagement, we did get better recruitment and better coverage in 2018. And this actually shows a GAM model looking at that concept of coverage, which is essentially the density of the AGO trap in these communities. When we had low density of these AGOs, we did not see evidence of suppression of female Aegypti. In fact, it might have been an increase in the abundance. But when we did have higher coverage rates, we were starting to see suppression that maybe translates to about 57% reductions. So that just highlights the story of that AGO intervention and that manuscript's in review. So now transitioning to the Harris County project. And again, this is a partnership thanks to a strong program that Harris County has had for many years, gathering a lot of good data. And we've already worked with them to publish some of this. So we looked at the how weather influences Culex abundance and infection with West Nile. But now we're addressing this question about how adulticide use over the years has influenced mosquito abundance and control. So here's a highlight of the massive data set. So roughly from 2002 to 2020, there's over 16 million mosquitoes that were processed, mostly are females. About 6 million pools were tested for West Nile. Over 5,000 of those were positive. Obviously, these are all mosquitoes combined. For the analysis, we'll be focusing on Culex quinquefasciatus, the main vector of West Nile. And then the adulticide data is interesting and also very large. So basically, whenever a operational area, we've already heard and seen maps of what those operational areas look like. Whenever there is a positive event, whether that's a mosquito pool or a dead bird or maybe a human case, that triggers a very quick response by the vector control group, and they deploy a truck ULV application of adulticide. They've used a variety of products over the years, but basically trucks get deployed to that operational area, and their goal is to treat it very quickly, maybe often the same day a positive pool is recorded. And then they treat it 3 times, that entire area 3 times over the course of a week. So that all adds up to, over this large time frame, there's been 39,000 spray events. So each spray event represents a truck in a given night. And then because these trucks are delivering a known product that covers a known area, which is essentially a buffer around the road, that allows them to calculate an acreage sprayed. So that adds up to 22 million acres sprayed over that time frame. So this is the kind of conceptual overview. And so this is, as you can imagine, a very complicated analysis. Luckily, Luis Chavez is a good collaborator helping with this. We have a new PhD student, Charlotte Rhodes, who's also helping. So the goal here is to, again, compare the adulticide data with the Culex abundance and infection, and again, broken down spatially by these units, which are the operational areas. And we're going to tackle these data with a variety of approaches. For example, an ANOVA that can compare the means before and after the spray event. But we also know that these models become very complicated because we know weather influences both Culex abundance and infection with West Nile. So those need to be included in these models as covariates. And in general, these statistical models of these really massive data sets have a lot of computational demand beyond what our own personal computers can handle. So that's partly been one of the delays in making progress on this analysis. but we now have all the data, so we're going to tackle it. So now I want to conclude with a cool and exciting project, this human survey project. Many of you have already been involved with helping with this and are aware of this. Katie Dickinson and Natalie Benakos are the leads of this. They're at Colorado. And so we basically wanted to ask the public, random people from the communities, what their willingness to pay for vector control would be, and then also what forms of vector control would they support. And so we targeted 3 counties that capture kind of a diverse demographics. That includes Hidalgo County, Harris County, and Tarrant County. And then luckily Qualtrics, the company, did their magic and helped us obtain 1,800 survey responses, which from the residents of these counties, which are very nicely matching to the demographics of the county. So here are some of the results for willingness to pay. So how much people would support paying for vector control per year. So roughly 15% of the respondents said 0. They don't want to pay any for vector control. 10% roughly said they would support less than $5. 50% would support $10, and so on. You can see a few people would support maybe a few $100s per year. And what's interesting is the breakdown of demographics in terms of this willingness to pay for vector control. So females were less willing to support vector control compared to males. People who had a graduate degree were more willing to support vector control. People who had a higher income were more willing to support vector control. And then interestingly, liberals were more willing to support vector control compared to conservatives. And then we also asked, among all the vector control tools out there, which ones would they support or be opposed to? So these included adulticides, larvicides, various forms of lethal traps, and then the various kinds of vector control that involved the release of mosquitoes. So this could be sterile insect technique, genetically modified mosquitoes, or Wolbachia infected mosquitoes. So overall, the support was pretty high. So this is a rank from 1 is strongly opposed to 5 is strongly supportive. And so overall, they're all above 3 for every form of support. These forms tended to have a little bit more support. So the lethal traps actually had the highest support. And then the genetically modified mosquitoes had the lowest support. Another interesting observation, we all know that, in Harris County, adulticide, including aerial adulticiding, is pretty routinely done. Whereas in Tarrant County, you'll hear tomorrow from Nina, it's not as routine. And so it's interesting to see that people in Harris County generally supported adulticiding more than people from Tarrant County. And again, this kind of shows the demographics of how people supported these different control options. And again, people with higher income supported these approaches a little bit more. And one thing that came out in both the willingness to pay and this one is that when people knew someone that had West Nile or dengue or Zika, they generally were more supportive of either paying or the control approach.
Gabe, you're at the end of your time.
Okay. So these are the conclusions of that human survey. And so, yeah, here are all the acknowledgements of everybody involved. Thank you.
Thank you, Dr. Hamer. Next up, we have Tom Shelite to close out this session before the live Q&A.
Good morning. Can you hear me?
Yes,
All right,
and we can see
great.
your screen.
Cool. All right, so we're going to switch gears a little bit from surveillance and everything to diagnostics development. So we're looking at rapid point of care nucleic acid amplification tests for basically every virus that we've been talking about up until this point, including a couple that we haven't really talked about. So the idea is diagnosis and surveillance of arboviruses. Currently, we just recently published a paper on Heartland virus, which is a tick-borne infectious disease transmitted by Amblyomma americanum, which causes a febrile illness that's potentially lethal, and especially in immunocompromised elderly patients, found throughout the Midwest to the East Coast. We use this as kind of a basically like a case study development assay, because we had access to what few patient samples that exist, and so we used it as basically as our poster child for developing our assays while we were developing some of our other arboviral tests, including with our previous work we've done with parasites. So we have some experience with developing RPA lateral flows. The overall objective is to develop these tests so that they can be used in smaller laboratories, so they don't have to send samples to larger regional laboratories and wait days to weeks to get results back, both here in the US as well as developing countries. Obviously, the goal is to develop these assays for Zika, chikungunya, dengue, and of course West Nile, and then eventually other and emerging new viruses, that anything that pops up, and then move into as well as detecting these viruses and their potential vectors. Over the overview of how this assay works, we have a master mix that contains a recombinase single-strand DNA binding protein, a strand splicing polymerase, and then we add our specific forward and reverse primers. The reverse primer contains biotin label, and then a probe, a FAM-labeled probe. We then you can basically take any sample type and extract the DNA or RNA. The RNA assays would require an additional cDNA synthesis step. Then you add this into the master mix that contains your probe and primers, amplify, and then during the amplification step, the probe will displace the double-stranded DNA, and then get the NFO enzyme will excise basically the blocking portion of the probe, and then allow for additional priming. And what you'll end up with is a dual label product, amplification product, that has both FAM and biotin. And then at the end of your reaction, you'll then dilute that sample and then add it to a lateral flow strip, which has antibodies both for the FAM probe and the biotinylated primer. And as it flows up the lateral flow strip, the FAM labeled end of the probe will bind to gold particles, and then the biotin labeled part of the primer will be immobilized on the lateral flow strip, and resulting in something that you see on the right of your screen, something similar. And so the variation intensity of the bands is typically due to the relative concentration of the amplification product, and that's usually a result of the concentration of your input. So, i.e., the concentration of the nucleic acid that's available in your sample. So this is where we stand currently with our assays between Zika, chikungunya, West Nile, dengue, and the recently accepted Heartland manuscript. So for Zika, Chick, and West Nile, we have the ability to detect 1 to 3 PFU equivalents per reaction and of cDNA in human serum. And then with dengue, it's a little all over the place, depending on which serotype we're detecting. Now, these are all for West Nile and Chick and Zika are very similar to the current assays that are the current real-time assays used by the cDNA or by the CDC. Sorry, not cDNA. Dengue 2 and 3 are pretty much spot on to the real-time PCR, whereas 1 and 4 are a little bit less than this, the real-time PCR. Our detection in mosquitoes for Zika and chikungunya, we're having a little trouble with inhibition, and that's something we're trying to work on. I'm not sure why that's happening. So this is just an example of what it looks like when we're doing clinical samples. I'll go back real quick. So that's the key problem with our assays right now, is that we're unable to move forward with publication for Zika and chikungunya, is that we're lacking in clinical samples. That's something I'm going to address shortly. What clinical samples I have got the test for Zika is basically what we're showing here. We had 8 samples that were suspected of Zika, 3 of which turned out to be positive, and the other 5 were negative. And this goes corroborated by real-time PCR. And this is just an example of our dengue limited detection for dengue 3. And then this is basically the developmental steps for West Nile virus. And this is pretty much how we set up everything, how we set up our RPAs. We start out with a couple, 2 or 3 samples of primer sets. And we do what's called the RPA basic, which is just our RPA parameters, testing different temperatures, looking at different strains of viruses, running it on 2% agarose gels to see which primer pairs are going to work or not work. And then after we determine which pairs work the best, we design probes and then move on to test them in the NFO steps. So for West Nile, we had a couple pairs that we designed. As we named, I named arbitrarily Vasquez 2016 and a Kumar Cyber, and ran them at 42 degrees Celsius with or without betaine to inhibit non-specific binding. And as you can see, the Vasquez kind of worked, whereas the Kumar worked, but without betaine, the Kumar had a non-specific band on the Viros. And then at 43 degrees, the Kumar still worked pretty well, but the Vasquez pretty much didn't work at all. So that's the pair of primers we proceeded with and designed a pair of probes. One probe I designed a degenerative probe, which had about 6 degenerate bases throughout the length of the probe, which, as you can see, and just using just water without betaine, has a non-specific binding. But once I added betaine, that non-specificity went away. But on probe 2, which had the identical sequence to the West Nile sequence that I was using to design the probe, it worked, had no non-specific binding. So we then proceeded to do a limit of detection using virus spiked in serum, and that's when we determined that we had a 3.13 PFU equivalent per reaction in human serum with no non-specific bands. So one thing we've ran into since the advent of COVID is that the company TwistDX is having issues with their NFO production, NFO kit production. For whatever reason, they haven't been upfront with this, and so about midway through production or doing this West Nile virus assay, we ran out of kits, and so we've been trying to figure out ways to work around that problem without having to create our own entire new master mix and everything like that. And so one way we determined was using their basic kit and adding an NFO enzyme into it, with the hopes that would make a simplified assay. And luckily, that turns out to be the case. With the addition of betaine, we were able to have a clean reaction at 42 degrees Celsius for the West Nile. I'm in the process of ordering more basic kits, and so I can test my other assays to make sure that this is a viable option. So that way I don't have to start from scratch. So for the future activities within our groups, with the other diagnostic subgroups, we're having multiple meetings to help with, as I say in step one, is streamlining acquisition of relevant clinical samples for each of our groups, which has been facilitated by Doctors Baralt and Munoz, because each of us require different types of samples. And then also with the help of Dr. Wilson's group, troubleshooting our molecular approaches, as well as developing ways to assemble lateral flow strips, as well as developing a digital reader for our lateral flow strips, so that we don't have any subjectivity of reading the strips. As you saw in the couple previous slides, that the really faint lateral flow strips, they get a little more difficult to read. And so having a digital reader that can have some type of OD reading could prove beneficial, especially for inexperienced readers. And then, of course, establish closer interaction between the groups. Future plans is, as I just stated, clinical samples is the main thing that's slowing down the CHIC and the Zika RPA validations. That's the only thing left to do, is to run 20 to 40 clinical samples, and I can send that out for publication. And then assessment of the dengue RPA. I'm looking at several other primer sets that I want to try to reassess to see if I can improve that RPA to make it better. And then also for to make the RPAs more useful is to do we call dual band RPA-LF, which by having the probes, one probe with a FAM label and another one with another fluorophore that we can use, so we can detect two different arboviruses at the same time. And then figure out the problem that I'm having with the mosquito and tick pools with the inhibition of our isothermal assays. So, and that would be it.
All right. Thank you, Dr. Shelite. We will now begin our Q&A session for our first four presenters, Dr. Pietrantonio, Dr. Slotman, Dr. Hamer, and Dr. Shelite. Dr. Roundy, do we have any questions from the audience?
No questions yet. Oh, we got another one from Scott. Dr. Weaver?
Hi, Gabe, I have a question for you about your survey information. Do you know, of the 3 counties you surveyed, what the mosquito control budget per capita is and how those numbers compare with your data on how much people are willing to spend?
Yeah, that was the slide I skipped over due to time. It was on my summary slide, so I'll just read it off to you guys. And this was all, a lot of you were involved in the interviews. So Katie and Natalie, they interviewed people from Harris County, Hidalgo, Tarrant. Many of you are on the line. So a lot of that background homework went into the development of that survey questionnaire. So the background homework delivered the kind of current status quo, which is kind of what helped design the question. So basically for Harris County, it's $2 per person per year, roughly. For Tarrant County, it's 25 cents per person per year. And then for Hidalgo County, it's 5 cents per person per year. That's roughly what funds organized vector control in each of those counties. And then the survey results suggest support is pretty high, closer to maybe $40 per household per year. So very different than what the current tax, how it works. Does that help what you asked?
Yeah, that's exactly what I was interested in. I wonder if any of the vector control folks in the audience would comment about how the politicians who are developing your budgets are thinking about this as far as expense per capita, value of the investment compared to the way people are answering the question.
While we give those guys a minute to send in answers, we do have a question from Dr. Aaron Brolt. Dr. Roundy, if you'd like to read off his question. I think you're muted, sorry.
Thank you. Aaron had a question for Tom. He asks, have you assessed quantitative capabilities of your assay with scanning of the LF strips?
Not yet. That's part of the work that we're trying to get. We were trying to get started with Dr. Wilson's group right about the time COVID took off. And so we're trying to get that started back up whenever we can get, more in touch with that group up there in Houston.
If I could add something, a bunch of stuff from my lab is now in my postdoc's garage. And so we do have some limited capability, and UH is more open now. So we're making some progress. We're scheduling a meeting now about this.
Yeah, we were really gearing up to do some of the stuff with Leishmania as our kind of our surrogate, because we have all that the DNA and everything in their hands right now, and that kind of the COVID kind of nipped that in the bud real quick. But as soon as we can, we're gonna start doing that.
All
I
right.
actually had a follow-up question for Gabe as well. Is there interest or plans to continue monitoring these attitudes towards different vector controls? I think that would be very interesting as more of the Wolbachia and genetically modified tools are rolled out throughout the US.
I think there is a lot of temporal variation in the response. And it took us like a couple of years to get this organized. And, Katie did a lot of background. She actually did interviews in person in each of the counties, and then a lot of the phone interviews. And then when we finally had everything together, Qualtrics helped us execute this last fall. And actually that was a relatively slow year for West Nile. So then it's like, I think the moment these surveys go out, I think perspectives and attitudes do vary a lot. And a lot of it, of course, is probably driven by media. Unfortunately, I think so much went into that. We need to just kind of focus on publishing what we have and then, consider what to do in the future. But certainly as part of our current center, we're not going to attempt to repeat that survey. But yeah.
Do we have any other questions for any of our presenters from this session? Oh, we have a question from Dr. Bruno Travi for Gabe. He asks, is Wolbachia used and how does it compare with insecticides?
Well, so yeah, Harris County could speak on this better than me. They did do a trial, I guess it was last year, working with Mosquito Mate, and the CDC was involved, where they did releases of Wolbachia infected mosquitoes in Harris County. So I guess it's a relevant control tool to ask the public about their support, but I'm not sure how it compares to in terms of their results. I think actually I was just in Harris County, I think it was last week. they were planning on going to the Texas or the American Mosquito Control Association meeting to present some of those results. And obviously AMCA was canceled due to COVID. So I actually don't even know how those Wolbachia trials in Harris County, how they worked out.
We'll also be hearing from Harris County tomorrow from Chris Frederigill, so maybe he'll touch on that as well.
Perfect.